Hello and good evening to everyone. Thank you for joining me as we, as usual, continue this journey of understanding the COVID-19 pandemic and speaking to researchers and scientists across the world. This is a very important topic and actually one that was raised from about August of 2023 with regards to circulating spike protein after vaccination. What we've got with us is Dr. Carlo Brogna and Joachim Gerlach, who are here to talk about some of it. Now, sadly, because it's Christmas time, but even though in this period of time we still have the commitment from our researchers, Carlo has only been given limited time by his wife to speak. So we will try and catch as much as we can out of him in that period of time. So let's just quickly get into it. We've got Carlo and we've got Joachim here. So Carlo, so we know your time is limited. I'd like you to just quickly introduce yourself to the audience, and then I'll ask Joachim to do a truncated introduction, and then we will be able to talk afterwards. So go ahead, Carlo.
Good evening to everyone. Thank you. Okay, it's a pleasure to stay here in this moment. I am Carlo Brogna, a physician, medical doctor, and oral surgery from Italy, and I'm a chief director of our facility, Cranium Group. So our line of research is based on SARS-CoV-2, and we have focused our attention on bacteriophage behavior of SARS-CoV-2 and toxin of SARS-CoV-2, and we also studied the spike protein effect of the mRNA vaccine. Okay, so this is my introduction, Philip.
Wonderful, wonderful. And Joachim?
Yes, good evening, good afternoon, everybody. My name is Joachim Gerlach. I speak from Germany. I'm a COVID-19 researcher, and I'm head of research and development for medicinals. And we are trying to develop products and protocols that can help people affected by long COVID or COVID conditions and spike protein from the vaccine.
Excellent, excellent. Wonderful. So, Carlos, since we know that you have some limited time, I think we want to start off with a very simple question. Now, you have done research looking at circulating spike protein after vaccination. Now, we were told quite clearly that that kind of thinking was misinformation. What made you decide to do the research anyway? What was it about it?
Oh, okay. So, first of all, we decided to make this line of research to better understand the half-life period of spike derived from mRNA vaccine. So we are curious to know if first of all the mRNA and spike protein will stay in the muscle here, okay, in the lymph node here, or go in the circulation. First of all, this is the first question of our paper. The second question is to understand for how many times the spike protein derived from mRNA vaccine will stay in the circulation, and to better understand the limit, in which sense someone could ask. The limit to understand if this spike protein grow up the antibodies, and if these antibodies protect or help the person to don't have infection of SARS-CoV-2. And we have discovered, every one of us have understand that the titer of antibodies is not sure to don't have the infection of SARS-CoV-2. And many persons that have titer antibodies high at the same time go infected by SARS-CoV-2. And we also wanted to understand how I just said before, the real half time of the spike protein, because if the spike protein stay for many times, 1 month, 2 months, 3 months, 4 months, 6 months before the booster dose, it is important to know, because if I have a spike protein and I have antibody level high or low, sometimes maybe the spike protein is read from our body as a self and non-self protein. So this is a very important question to understand, and this is the why, because we have made this line of research. In this period, we are calling all the patients that we have make the first paper, and we have try again to do another experiment to understand if the spike protein will stay also after 6 months. But you will discover in the next paper this data.
Wonderful. So we've got here the paper here, and this was looking at the detection of recombinant spike protein in the blood of individuals vaccinated against SARS-CoV-2, possible molecular mechanisms. So before you have to leave, I'd want to ask you your thoughts about it. But just in case people want to know, the critical thing that they discovered was that the spike fragment was found in 50% of the biological samples analyzed, and its presence was independent of the antibody titer. But this was a small study. This was only about 20 people who were vaccinated. Can you extrapolate on that kind of small study? Any thoughts on that, Carlo?
Sorry, I have a problem with audio. I don't understand.
Joachim, can you just explain that for me in Italian?
Él solo tenía 20 personas en este estudio, y si se puede extrapolar esto, ¿cómo se puede extrapolar esto a un grupo más grande? ¿Puedo preguntar en italiano?
Ok, ho capito.
Extrapolar es como...
I understand. I can answer.
Yeah.
Okay. I understand. So in the first during the pandemic, we have selected only the medical doctor who make obligatory the mRNA vaccine, and we make only 20 doctors because no many want to participate to the study. In this moment, there is also we have some difficulty to enroll many persons, because in Italy there is a very down the percentage of person who make the booster dose in this period. So we have a very difficulty to enroll the person. Okay, I think that the good windows to make this study would be at the first of pandemic with the first injection, but we have not developed yet this method to do this research. So now it is limited for this reason. So the reason is that the method was developed in the last part of pandemic, and only 20 doctors would to participate to the study. Now we have the problem that we no found person who make the mRNA vaccine, because it's the level of vaccination is very down in this period after pandemic. But I think that the same study could be on the animals, could be performed on the animals. So if we make some fit back, I think that before to introduce a new technology in the industry and for the large utilize in the person, is better to experiment on the animals the effect of the drugs. So the next time I hope that many other scientists and doctors could make this type of experiment before inject the new drugs with the new technologies in the person.
Yeah, important point. Joachim, I'll come to you shortly, but I just wanted to pull up a section in the paper here for Carlo, because as I said, I know his time is limited. Now, in this part of the paper here, one of the questions that people would wonder is, how did you know that the circulating protein wasn't from infection as opposed to vaccination. And so I was trying to understand, are you able to expand on that? How did you tell the difference between the spike protein from a vaccine versus
Yes.
the spike protein from infection?
Yes. With the proteomic, with mass spectrometry, it is possible to discriminate the two type of spike protein, and we have only selected people that have the spike protein of mRNA vaccine, and we have focused on this type of fragment that have the double PP, okay, the double PP, and only the doctors who give the blood, and we have sure that they have no exposure to SARS-CoV-2, and that they are not infected of SARS-CoV-2, and we have make the test that in the blood they have no spike of SARS-CoV-2. So we have only selected the person who have spike with mRNA vaccine, and the method is also under patent in Italy. This is a method that in Italy we have make a patent on this aspect. So what is important to state that some of doctors who participate to the study have no antibody level, even if they have a spike of mRNA vaccine. So not always the spike of mRNA vaccine generate antibody. So this is another state of the paper.
So are you saying that you had situations where someone had circulating spike protein but didn't generate antibody?
Yes.
Wow, that's really, really interesting.
And
Okay.
this is the reason because we say that it is independent from the antibody titer in the paper. We make a phrases on this argument.
Wow. And so in terms of, again, coming back to the paper here, it shows here that in this section here, after vaccination, this is looking at the amount of circulating spike protein, which is up to how long? What is the duration of time that you are able to see circulating spike protein?
We make a picture. So we have selected the physician and doctor who make the mRNA vaccine, and we have make a picture before they repeat the booster dose. So the picture it is an images in a moment. This study, and in this moment we have focused the attention. If you give the blood in the samples for the analyze in the study, if you have the spike of vaccine or the spike of the virus. If you have a spike of virus, you are discharged, and if you have the spike of vaccine, you are enrollable for the study. We have our windows is in the first 6 months after the complete the mRNA double dose and before the booster dose, because if we enrolled the person with the booster dose, it is sure to find the spike after the booster dose. So we have make a picture from the second month before the booster dose to 6 months.
Mm-hmm. Yes, okay. And I know time is short for you, Carlos. I want to ask you one other section here before you go.
Okay.
And that's when you are thinking about in the paper, what could be the mechanism of this?
Yes.
And you have 3 different ideas, which Joachim and I will talk about afterwards, but I just wanted to hear your thoughts. The first one being that the spike protein is integrated in cells.
Okay.
The second one being that the spike protein is being produced with the pseudouridine afterwards,
Okay.
and the third is to do with bacteria.
Okay.
Could you just generally explain to the audience what your thinking is on this?
Okay, now we are testing these 3 hypotheses. Okay, and in the next paper, all of you will read the result. To explain the first hypothesis, if there is an integration of the cells in the cells, we are recalled all the person that we have called in this study. So after 2 years, we will perform the same experiment, and we will know if the spike protein is still present or not. If the spike protein will be not present, so we must discharge the first hypothesis because there isn't an integration in the cell. The second hypothesis, it is a consequence of the exclusion of the first and the third hypothesis. And we also now will perform also the third hypothesis. We are collected the blood samples of person who make the fifth dose of booster vaccine with the spike protein. And now we make the hemoblood culture of bacteria, and we will extrapolate, we will select the bacteria, and we will analyze if this bacteria will produce the spike protein because they are contaminated with mRNA vaccine. So if the first or the third hypothesis will not work, we will think that the second hypothesis is the most accreditable. So a very long constitutional production of spike protein for a long term, but non-integration in the cells. So.
Wonderful, wonderful. Joachim, I know I wanted to ask you if you had a question for Carlo before he had to run back to his family commitments.
Yeah, you just posted the three main questions that were already on my mind. So for the moment, I'm fine. One question is, of course, the lack of antibody presence in the presence of spike protein. Carlo, could it be that they are many of these spike proteins are inside extracellular vesicles? Because we have had new papers coming out in the last few days on extracellular vesicles or exosomes containing either virus and or spike protein. Even when the PCR tests were already negative, they could still find a lot of virus inside extracellular vesicles. And it's been documented now in many, many different papers, the presence of spike in EVs, and that might make it impossible for the antibodies to build up a reaction because they are kind of shielded inside of that.
Okay, yes. So, Joachim, I started to make this line of research on SARS-CoV-2 3 years ago. And we started to observe that the virus was replicated in bacteria, as you know. And we also have published a paper on MDPI, where we state that some protein of the virus have many D-amino acidic, and so there are in the blood of a person and derives from the bacteria. Because many proteins that have of the virus that have are abundant of D-amino acidic are produced by bacteria. Okay, so in this line of research, I have understand that many concepts of the old virology aren't right. So first of all, we must understand that not always who make positive to PCR, RT-PCR, could have the antibody against SARS-CoV-2. And sometimes we have the opposite situation, that many persons who have no PCR test have antibody against SARS-CoV-2. So I believe that we must make some major study to understand better the mechanism of production of antibodies and of link of the SARS-CoV-2 to bacteria first, and then after bacteria to the epithelial cell. The argument is very many big, and it is we know very few about the real mechanism that can occur in the reality. Okay.
So it is a possibility that the lipid nanoparticles will deliver the mRNA into bacteria, and that the bacteria might be capable of producing spike protein. Is that
we are testing this now. I don't know if I don't make the test. I can say nothing. But if I will found something, I will publish. Okay.
Okay. Yeah, because
Excellent.
very short, one more thing, because we see in many of the vaccine injured persons, we see the same dysbiosis picture as in long COVID patients. As the lack of bifidolactobacteria and many other Akkermansia and Prausnitzi that is usually after COVID infection is also being observed in the vaccine injured biomes of biomic testing. So something speaks for your hypothesis that this is at least theoretically within possible.
Okay.
Yeah.
My point of view. Do you know we have a colleague, we have a speaker many times on phone. So Joachim, you know my point of view. I have observed that also the person that don't make the mRNA vaccine stay not very well after infection, and they have long COVID condition, and many other person also die even if they make the mRNA vaccine after infection. So we must be honest in every aspect. I believe that our study on the toxin level produced by the bacteria is underestimated. All the patients that we have treated with the double solution of antibiotics, amoxicillin and rifaximin in particular, have no long COVID condition, and they stay well. They have no problem, and I believe that the secret is in the microbiome. I believe that the antibody level against SARS-CoV-2, even if they are produced by vaccine or by a infection obtained before, they can stress the bacteria because they repulse the SARS-CoV-2 always against the bacteria. So it is very underestimated, I believe, the problem of microbiome and how many important is microbiome to generate the stress of our body.
Excellent, excellent. Now, I am very much appreciative of your wife, Carlo, giving us some time, and I don't want to get on her wrong side. So we really appreciate you jumping in on this this holiday period with your family to share some of these ideas.
I am sorry.
No, it's fine. It's fine. We appreciate the time. So I'm sure we'll have a chance to speak again soon, Carlo. Myself and Joachim will continue to discuss the paper for a little bit longer.
Okay, thank you. Thank you.
Thank you very much.
Thank you so much. Grazie mille.
Happy New Year's to everyone.
Ciao.
So yes, Joachim, it raises some important questions. As I said, that it goes back to the original point of assessment. Now, Joachim, you're in the business of having to go through testing to bring products to market. Can you see how this could occur?
Yeah, of course, it's always been blamed on the hurry and warp speed and no time and pandemic pressure, etc., etc. So, look, what can I say? We see now the results of all that in the patients. So after some years, so it is without any question, it was a very unprofessional approach, to say the least, if to introduce a product like that. I mean, we're working mainly with not pharmaceuticals, with dietary supplements, but even there, we go through extensive preclinical trials and even clinical trials. So this could have be done better. The problem is that the idea of persistent spike production after vaccination outside the deltoid muscle was from the beginning on a complete misinformation, and friends of ours like Dr. Stephanie Seneff and others were heavily criticized for even bringing up a point that would suggest something like that is happening. So I think that the industry should now be more quiet and do exactly what Carlo and our teams are doing, and look at what is happening out there in the patients, because a prolonged production of spike protein is not the initial idea behind the design of this vaccine, isn't it? And so the questions are from our side. You know me now for quite some years, and I don't look at blaming it on anybody. I just look at what is being broken, that it must be fixed. So our next challenge, again, like always, is now a new challenge. What can be done about that? Which cells are that? Why are they producing spike for such a long time? And we heard now 3 different hypotheses. And maybe it's also interesting to hear your take on what you think is the most likely one. And because if you give me now 3 different drug targets, it's going to make it harder for us.
Yeah, it's an important question, and I remember when I was looking at long COVID, and what's interesting in what Carlo was saying is that these people didn't have long COVID; they were just vaccinated. Now, in the research that we've been looking at around what happens in long COVID, when you looked at the biopsies, certainly the gut biopsies, you could find fragments of the protein in the gut epithelium, CD8 cells, as well as some of the epithelial cells in the intestine. And this was up to 100, 200 days after the infection. So this is about infection, not necessarily vaccination. So the question is, where is it that this mRNA is still being active? And in my view, when I reflect on the time frames, if this is going on up to 187 days, it's unlikely to be an epithelial cell because they die quite quickly. And so it suggests that this could be immune cells if it's not bacteria. So I think the bacteria certainly is a possibility, but that's an even more complex jump to try and understand where spike protein could be being made for such a long period of time. One of the questions I didn't get from him was whether or not spike protein was it full spike protein or was it pieces of spike protein? I didn't
It
get to
was
ask.
full spike. And okay, they didn't do electron microscopy. So what they found was the proline sequence up and down from that. So that is let's say, okay, then actually it's a sequence of the spike that they found, because they degraded the spike from the vaccine with trypsin, and then the degradation process will result in a different sniplet, different epitope. If you do that on a vaccine spike than it does on a wild type spike. That's how they were able to distinguish between wild type and vaccine spike. So no, it was not the whole spike. They just, sorry, I have to correct myself. So it was just like that particular insert, that particular epitope that they were detecting. But that's enough to give away that must be spike production ongoing, because otherwise you wouldn't find just this particular giveaway in
And
the
so this is part of the question. Now, they were looking at a specific epitope. Is it possible, therefore, that other epitopes, this is why I said, is it a full spike? Is it fragments of the spike? Could there have been even other bits of spike that were circulating if we were looking for them?
Now, look, from our work, we are looking at how to get that out of the organism, and we are always distinguishing between 2 different persistent models. One is the productive model, which you have with viral persistence, and now Carlo added ongoing spike production after vaccination, and then you have the let's say the non-productive persistence. That means that you have spike protein either from wild type or from vaccine spike persisting either on cell membranes or inside of cells for a long time. And so in this case, look, if you find the fingerprint and in the hand of a terrorist, then you don't have to wonder if there were terrorists around when that fragment was detected. Yeah, so it's a very clear indication that you do have existing spike protein in the serum when you can degrade them with an enzyme, and your leftover is exactly the giveaway epitope that is very specific for the vaccine spike protein. So I think it holds water that we are looking at ongoing spike protein production from transfected cells. Now the question is which cells, but mainly for me is the question, what does make these cells not die or go into apoptosis, or is the mRNA not used up in the process? So, or is it even producing a spike protein within bacteria, which is a very exotic kind of sort? Now that if you have to imagine that there is a bacteria now producing spike proteins from the vaccination, the LNPs going into a bacteria and being able to recalibrate the internal mechanisms of the bacteria to start reading mRNA and producing spike proteins within a bacteria. That would be a complete sensation, in my opinion.
Well, I think that the more that you reflect on it, now I think that this is part of the problem is we've never seen a virus that has the capability to infect bacteria. That's what we call a bacteriophage. They don't usually infect humans. They'll only infect bacteria, and human viruses will only infect humans, or you'll have animal viruses and so on. So this capability is quite unusual. We'll not go into the origins at the moment. We'll just focus on the science a little bit. But what it indicates is that the virus, if it is turning bacteria into the, if it's targeting bacteria where bacteria turn on us, that's a horrific circumstance because we only exist because the bacteria are living in harmony. It's like a what we'd call a black piece. The bacteria get from us, we get from the bacteria. We live in harmony. But if we turn those bacteria against us, the outcomes are just horrific.
Yeah, I mean, we at the moment this question will be settled by Carlo. They are working on it, so we will find out soon enough. Where does the spike come from? Yeah, we can now only say we detected, or he has detected it even after 6 months after vaccination, which is not a good thing to have. And of course, you have to understand one thing: the spike protein is something that has a very high affinity to fuse with any cell that it crosses. And so if you find a lot of free spike floating around in the blood, you can bet on it that there's more spike that has already fused with the cell membrane, is either on the membrane or inside the cell, and stays there for a long time. Yeah, and like I said, this deposit-like persistence of spike. So it's a warning sign. If you find free spike, that means that you have at least 10 times the amount already being deposited into cells. It's like you have a magnet on a string running through a metal pipe. Many of those magnets will not reach the bottom. They will stick to the wall somewhere, like the spike protein. It has a super high affinity to fuse with heparin sulfate and all the cell receptors. I mean, we've been talking about that for ages now. So that is another big problem. And so if we say it's within human cells, then it leaves the hypothesis that was brought up between me and Stephanie like a year and a half ago with the line 1 and even other reverse transcribing methods that could happen with a spike protein, that it could integrate into our DNA even. And now lately we have seen the SV40 promoter that can also assist in the integration into cells, and that might lead to a ongoing production of spike protein. I don't know exactly, but
What I just wanted to say is because one of the questions, and I remember this being raised by the proponents who were saying, say, for instance, with SV40 or DNA fragments, they were making the point is there is no evidence that those things cause disease, and in the same way, it could be argued, even though we are seeing circulating spike protein, it doesn't mean necessarily that it is causing disease. How would you respond to that?
I would say then go back and do your homework. So if you look at what spike protein can do and has proven to do, there is not a single question that spike protein by itself and or SARS-CoV-2 as a virus can cause tremendous harm in the body. It doesn't have to happen in everybody, but it's like you would say, you can also have some free floating asbestos in your body, or uranium, or you can smoke 50 cigarettes a day and that's no problem. Yeah, it will be a problem in some people. Other people might not have a problem. So I would say that the spike protein by itself is a pathogen, is a really dangerous, wicked protein, and it depends where you have it in your body, and what is your predisposition, and what is your state and your age, and God knows which other factors play into it, but that it can do tremendous harm, causing immunodeficiency, is oncogenic, it's neurotropic, it's causing metabolic disorders. It can get into the immune cells, like you mentioned before, as a virus and or spike protein. It can cause, let's say, a qualitative decrease in your immune responses, and the list is endless. I mean, we've been doing nothing else now, you and I, and many of our colleagues, for almost now 4 years. So to say that the spike protein is not dangerous is a gross underestimation, and it's not really true. If you do
Yeah,
your
no,
homework, you'll find out. There's enough
I,
papers
I
out.
I agree. I know. Here is just me sharing some of my thoughts on this. Now, I've brought back up the picture here to show you the spike protein here. Part of what our research very early on in the pandemic was that the spike protein from the virus, if it bound to ACE2, if it attached onto here, then the body could run into problems differentiating the ACE2 attached, free ACE2 attached to the spike protein, and therefore make antibodies to both. There are other proteins that bind to the spike protein. And so, from an autoimmune perspective, to have circulating free spike protein in the bloodstream for an extended period of time, which could potentially bind to other serum proteins where this is highly immunogenic, would almost certainly drive an autoimmune kind of picture. And I think that's really, from my perspective, what I've always been very worried about is that if we're having this kind of pattern where the immune system has been highly activated to target spike protein, and you have a situation where spike protein is circulating, almost invariably it's going to drive autoimmune disease. But sadly, that takes a long time to present.
Yeah, what we see now in many long haulers is now let's say the early cohorts from 2020, that were in that had the bad variants. Let's put it that way, the more aggressive variants. If they go untreated for a long time, or are treated in the wrong way, we now see that some of them are dying right now, and so, and it's really sad to see. And in our opinion, it is also now they turn more and more into an autoimmune state. Now, the longer this goes on, and the longer you present spike proteins or viral fragments on your cell membranes, the higher the chance that you develop autoantibodies that will also go against your own cells. And we see that especially in the central nervous system with these people, brainstem and other areas, and they really die a very unpleasant death. They kind of, everything is paralyzed. They can't breathe anymore. It's almost like ALS kind of symptom, and it's autoimmune driven in the end, in the last stage.
Yeah, this is worrying. Luckily, I think it's small numbers at the moment in terms of not small numbers. Goodness, don't say that. The hope is that we can understand this. Now, in the last bit here, Joachim, let's try and see if we can exercise our minds again. Because at the end of the day, it's one thing to talk about what mistakes were made, what could have been done better. But the truth is, we are where we are now, is that a lot of people have had circulating spike protein, possibly. They may have these issues either in the background in the past. The first question is there any way to identify who is at risk? Because that's the first question. In all aspects of medicine, we usually have tests that will guide us to know who has been affected. So if you think of diabetes, we'll be looking for an HbA1c to see if it's elevated to show chronic levels of high glucose, and then you know this person is pre-diabetic. One of the problems is that we don't seem to have any clear markers outside of the symptomatic people with long COVID. Anything that you've come across that could guide us to who may be being affected?
This is the problem is that we remember from from Bruce Patterson's panels all the way to all the publications from Protrino and all the other long COVID centers and cohort studies, you see that there is a shift, and you cannot really grasp and say, okay, these are the markers that are relevant for long COVID or vaccine injuries. In the last 6 to 8 months, we have been gaining a lot of ground in the US with our partner, Dr. Robin Rose, and her colleagues. And so, yeah, there are some markers now that seem to be really prevalent in most of the long haulers that are vaccine injured or spike injured people. One of them is TGF beta. And unfortunately, it's a lot of neurological markers like the GFAP, glial fibril astrocyte proteins, and VEGF, amyloid beta 40-42 ratios, and these kind of things. And that's a little bit worrying because, and especially in the last two years, it's 2 years now with Omicron, which is more neurotropic. So I think that it's going to be very difficult to find a one-fits-all panel that you can determine now somebody is a long hauler or not. And many disease expressions you see now are not really specifically for long COVID or vaccine injuries. They are normal. I mean, you're a doctor. You have seen before myocarditis. You have seen strokes. You have seen cancer. You have seen all these kind of disease expressions. We just see more of them at the moment in numbers. And so even there it's difficult to nail it down and say it was spike protein or COVID. Now, because you just have anyhow normal, let's say normal disease expressions, but just much more of them. Yeah? So that makes it very hard to pin it down. But as far as we see now, let's say there's one marker that you can see in everybody, but you need a fluorescent microscope for that. That's like what Beate Jäger, Recia Pretorius, and now my colleagues in the US are doing. And that is, for me, one of the safest indicators that there is a problem. And that is these, sorry, I don't have a picture right now at hand, but it's the micro-clotting, how it's called. So you have different kind of clotting findings under the fluorescent microscopy. Some of it is endothelial debris, platelet aggregation, and real thrombi, small, very small. And both clinics now, Germany with Beate and in Connecticut, have problems to find normal blood. And it doesn't matter if anybody has symptoms or not, or is a long hauler or vaccinated. It doesn't really matter anymore. I mean, Robin called me just a month ago or 6 weeks ago, and she said, like, is there anything wrong with our equipment? And I said, no, let me ask Beate. And Beate said, look, we did the same analysis. We do that now since 2.5 years or more. And so in the beginning, we had lots of normal blood still available. And it's getting harder and harder and harder to find control clean blood without microthrombi or clotting in there. And that is a real warning sign.
Yeah, that is pretty serious, and this is what I'm saying to people, and this is tying into when I recently spoke with Gert, and Gert was talking about a new variant coming, and I was thinking to myself that from my point of view, from the autoimmune point of view, I think the combination of the two things, which is a highly infectious, more virulent variant that triggers an autoimmune response, which leads to more of this kind of pattern with micro-clotting, is a horrific challenge to come up against. So let's just imagine again. So this is what I said before we finish. I'm trying to see if I can think out solutions. So we're seeing this micro-clotting occurring. We're not clear on the mechanism. And one of the things is people have asked me about things like triple anticoagulation and so on. And I'm saying I hesitate to use that largely because you don't know how long you need to use it for. And there are risks with doing that. We need to find primary pathophysiology to understand what could be driving these microclots, to be able to address that. And then in the interim, if you were using a method to try and reduce the symptoms, that's different. But we are flying blind at this.
Not
It's
really, not completely, no. So let's say the major driver of micro-clotting, in my opinion, and some of my colleagues, is the persistent spike in the endothelium. And that will lead to an inflammatory condition, and it's feeding the coagulation cascade from many, many angles. I can name you like 40 different pathways how that can happen. And so if you can give anticoags, yes, you can give a lot of other even natural products that will hopefully bring down the microclotting. But if you want to really get it stopped at the source, you have to get the spike protein out of the endothelial cells.
mm,
And so the better part of the last several thousand hours, myself and other colleagues were dedicated to that alone. Because without removing the spike from these cells and other cells, you will never, ever get better. And it's not enough to throw some enzymes on the equation. It's like throwing some spaghetti to the wall and hope they stick. That's not going to work. You have to go and do a detailed research and experiments and look at what is available and then slowly calibrate protocols. So the last protocols were able to significantly and in a sustainable fashion reduce the microclotting and especially the endothelial debris part. Because you shouldn't forget that these microclottings, especially in the small vessels in the brain and in other organs, will of course cause a occlusions there. And this will mean that you have not enough oxygen in your brain and in other organs. Just one of the side effects that happens after that. And so again, coming back to Carlo's finding with the persistent spike protein after vaccination, as we, again, like I already said, we distinguish between two different kind of persistence, a productive persistence and a unproductive persistence, because the spike protein, once it gets into your cell, it will not reproduce, but it will stay there forever. Because as long as the cell lives, it turns these cells into senescent zombie cells, immortal, but also dysfunctional. And the same actually happens also in the productive cases. So if you have a viral persistence in a syncytia, for example, then these cells will send out certain signals, and these signals we use to attack them. It's similar like a surface-to-air missile station, and you fly over it, and then this station switches on its radar, and then you can knock it off because it has given away its position. So the idea is to take out the cells that contain spike and even the cells that produce it. So I'll fly to Carlo and we will do some experiments on these patients that he could find the spike on. And then we see if we can stop the production of that. And I'm very positively optimistic that this is possible. And so this is the main point we have to get to, is to take out that spike. But you cannot just knock it out and have it free floating in the body or in the intestines even, because as I said before, it has a high affinity. So you just take it out from let's say damaged cells, you bring it out there, and if you succeed, you need to bind it. You need to bind the spike in order to get it out of the organism in a safe way. Otherwise, it'll go to the next door cell and do the same again.
yeah. Listen,
It's
I
very
mean,
difficult.
this is complex stuff. Now, as we close out, Joachim, let me explain to people an important thing. There are many people who still argue, oh, we don't believe that, or we don't think this. What I think the public needs to understand, this is so complicated that if the whole weight of the scientific community is not devoting their attention to trying to find answers, this is an insurmountable climb. It really is a complex question. And what we need are people who are willing to step outside of their ideologies. Forget for a moment what they thought, who is right, who is wrong. Let's just think, okay, if this is occurring, if people are getting sick, how do we mitigate the damage? And from my point of view, is that what I am concerned about is that I don't think we can reverse what has happened, but I think we can mitigate the impact. And that's an important difference, because if this is going to cause mortality and morbidity, it will still occur. But if we're able to influence it even by 20 or 30%, that's still huge impact for populations in terms of protecting individuals. But I do think, similar to what Gert had said, is that the wave has already passed. We're now trying to save as many as we can. What are your final thoughts, Joachim?
And my final thoughts are, of course, that the problem is that the medical communities and the authorities seem to ignore the incredible findings of, for example, Carlo Brogna. Now, Dr. Carlo Brogna has done now 2 groundbreaking publications, and both were discoveries that nobody in the whole world have done. First of all, to prove that SARS-CoV-2 is infecting our own bacteria, which is a sensation, and it can be treated even with antibiotics. That's number one. Number two, now that we still find circulating spike protein after 6 months. So they should listen to our group and to you broadcasting, and they should learn. And if they don't understand something, then look it up or contact us. We are even helping people to understand what's happening. So of course, that if have you seen any hospital now administering amoxicillin or rifaximin to COVID patients in order to stop the viral propagation in the gut? No. Do you see now at the moment with this new wave a lot of people again on the respirator? Yes. Have they learned? No. Who has proven that this can be avoided completely with H1, H2 blocker and Montelukast? Dr. Shankara Chetty. So why are they not listening to the people that really know what they're doing and that have proven now in the last 3.5 years that they know what to do and what works? Why are we still, like in a treadmill, still in April 2020? Look at the hospitals right now. That's ridiculous.
Yeah, I agree with you. And the sad part is that history will not look at this period of time in a positive light. I think that I could understand questions, as you said, in 2020, even 2021. But the minute we started to see that our interventions were struggling to control the spread, we should have been going back to the drawing board to try and understand what was missed. And when I say we, I don't mean we, because we have done that, but I'm saying as a whole, the scientific community, scientific and medical community, should have been willing to stop, take check, look again, and make sure that they were not missing anything critical. We're in difficult times, and the reality is that I don't think there are any easy ways out, but we have got to try to see if a difference can be made. So thank you very much, Joachim. Thank you for getting Carlo. We appreciate that he was here with us for a short period of time in this Christmas period. And thank you as well, Joachim. And I wish you all the best with the research and developments that you guys are doing to try and help people to be better.
Thank you so much. Merry Christmas still to everybody that is still in the Christmas mode. And thank you so much, Philip, for all your work.
Okay,
Goodbye.
wonderful. Okay, bye-bye.
Thank you. Bye.